Good evening, everyone. This is Journal Club 18, Club for the Advanced Science Academy. Shannon Ragg is going to be presenting a paper from Brain, which is a fairly prestigious journal with a fairly high impact factor of almost 14. This is sleep deprivation impairs molecular clearance from the human brain. Eide et al. This is in Ringstad's lab, I believe, right? No, it's actually in Eide's lab, MD PhD in Oslo, Norway. Okay, Shannon.
Okay, so this is a paper. It's titled Sleep Deprivation Impairs Molecular Clearance from the Human Brain. So there's a lot of experimental data that suggests that sleep is required for the clearance of waste products from brain metabolism. And in this research, they studied the questions of, does one night of sleep deprivation affect molecular clearance from the human brain, and if clearance is affected by subsequent sleep? So in order to do that, they had two comparison groups. They had the sleep deprived group, which did not sleep from day 1 to 2, and then a sleep group, which was like the control where they could sleep without restrictions. And they were injected with an MRI contrast agent called gadoberstrol, which was administered and like ejected into their spinal canal. And then during a 3-day period, they had constant like MRI imaging scans being taken, and then they had like a follow-on up 4 weeks later. Would anybody like to volunteer for the first figure?
May I provide just a little more background?
Yeah, go ahead.
Go ahead and unshare your screen. I'm going to show something.
Okay. Why is it not?
Transition. So there is now some appreciable amount. I think you got to unshare your screen. There we go. I think now there is some appreciable amount of data about this, but I want you guys to just appreciate how crazy this is. We have known as humans for forever, pretty much, that sleep was important, right? If you are kept up and not allowed to sleep, you eventually die. I think the record is something like 11 days without REM sleep, and then you die. So we know it's extremely important. And the entire field of sleep research has been going for centuries. And we've made a lot of good strides. And if you're interested in sleep, at the end of this, I should probably mention Mary Carskaden's work at Brown, because she runs a program. I'll tell you about that later. Just remind me. But I was at Brown University when this paper was presented, and this thing may win the Nobel Prize for them for Niedergard eventually, because until 2013, we didn't know what sleep was for. Think about that, how crazy that is. We do not, until 2013, we knew it was important, but if you asked any expert on sleep why we needed it, they'd be like, I don't know, we just know that we do. And this paper, which was published in Science 2013, was a landmark paper, extremely important in establishing that there's this new amazing lymphatic system in the brain, which we didn't know about before. So the lymphatic system is flushing out all of the waste chemicals throughout the night. So sleep is helping it's doing all the psychiatric stuff, like reorganizing memories and re-encoding thoughts and feelings and all that stuff, and dreaming and all that. But its real importance for staying alive was that it was cleaning out the system. And then since then, there've been a number of papers about kind of nailing down what exactly is being flushed out. And Shannon's paper that she's presenting today is kind of the first time it's been done in humans. Okay, but I mean, like, just think about that. 2013, the freshmen were like -2 years old, right? So this isn't that long ago. 2000, what is that? That's like 9 years ago, We didn't know what sleep was for. We just knew it was important. All right, Shannon, you're good.
Okay, so would anybody like to volunteer to do figure 1?
Sure. So this is showing different like slices of the brain and demonstrating how they gave, I guess, the patients like tracers, which are markers that will show up on MRIs in their brain, and it shows you the concentration of the tracers and compares the two between the sleep deprived group and the group that was like freely given sleep. And the group A, which is the sleep deprived group, you can see that there's like a higher concentration around like the different parts where it's redder. So at the, I believe, like the frontal lobe and a couple other areas, when you compare it to the group that has sleep, it's a much higher concentration, meaning that the tracers weren't actually flushed out, presumably by the lack of sleep. Inside like the center, you can also see kind of like a speckling of like higher concentration areas, and the group C, which is the difference, like demonstrates which areas are most like greatly affected by the lack of flushing out of tracers. And so it's essentially the difference between the number of tracers of the sleep deprived group and the group that was allowed to sleep. And so it's really high. So like around like, I don't know what parts it is, but it's like where it's like red on group C is where there was the most tracers that were left inside the actual brain, presumably by the lack of sleep.
Yep. Johan, you have a question?
Yeah, exactly what is being traced?
I got that one. It's called gadolinium. So one version of gadolinium is the one that they're actually using here, which is well, we just call it GAD, but gadobutrol, I believe, gadobutrol. It's hard to say, but we just say gadolinium. Gadolinium is the old one that this is based on. So it's something that has I think a bigger dipole moment. Is Miss Patel still here? Have you guys talked about dipole moments? Miss Patel, if you're around.
Yes, we did cover that
Okay. And
in AP.
would a magnet affect that? A very large magnet?
Yes.
This is usually they usually do these studies on 3 Tesla magnets, but I actually didn't notice. Did anyone notice what magnet they used for the MRI? It's a 3 Tesla Phillips, right? That's the one that's usually used for these type of studies. So that's pretty powerful.
From personal experience, it's very large.
So I think I've told some of you this. The largest you guys, I know you're gonna make fun of me for this. The largest MRI machine, okay, the largest magnet in an MR east of the Mississippi is at Vanderbilt, and it's 7 Tesla. And I was used as a subject in that several times, and it is disorienting. Like, you think you're just going through magnetic field, like it's magnets, it's not going to affect you. Oh my goodness, it makes you so. It makes me, everybody I know who's done it, it makes you really disoriented. Almost to the point of like vomiting, like you're on a roller coaster. So really powerful.
Just laying in a regular MR machine makes you disoriented. I mean, aside from the fact that it's very close quarters, but the magnets themselves can make you dizzy. They can make you nauseous. It's just disorienting in itself.
Can someone orient us to what we're looking at? What plane we're looking at in the left column, middle column, and right column of the figure?
Not my radiologist.
Anybody? We've done this already. Nolan.
So in the left, that would be axial or superior. The middle is coronal, and the right is sagittal.
All right. And here's the tricky thing. For the left two columns, when you're looking at the right side of the brain, it's actually the left side because you're always looking up from the feet. And so also the middle one, when you're looking at the right side of the brain, you are actually looking at their left, if that makes sense. So another thing to consider when you're looking at this is that everybody's brain is slightly different. So if you look at the kind of ghostly image in the rest of that's not brain, do you see how smeared it is? It is like really kind of hazy looking, because they've actually gone and had to stretch out individual people's heads and brains so that they would actually fit onto a model, which is the way that we do it, and that's perfectly fine. Cool. Thank you, Nolan and RC.
Okay, so we'll go to figure 2. So figure 2 is similar to the previous figure when it comes to like the brain pictures or the MRI scans. The MRI scans show similar trace enrichment in both groups at the end of the first day before sleep or their sleep deprivation in the cerebral cortex of the gray matter and the cerebral white matter. So that's like shown in A and B. And then for C and D, it compares a tracer administered before the tracer is administered, which is the blue, and then the normalized T signal has increased after it's administered, like afterwards, within the gray matter and the cerebral cortex and the cerebral white matter. Okay, so that's figure 2. And then would anybody like to do figure 3? Okay, Nolan, you can explain it.
All right, so this is a brain that was in the sleep group, and it shows the tracer enrichment as it's changing over time. So it shows from those 3 different views, and throughout the first day, you show how it is increasing, and then at 48 hours, it's begun to decrease. With sleep, it's mimicking how there's molecular clearance after sleep, and then the last one is when they checked back at 4 weeks, and it shows how there's like not a significant amount.
Okay. Yep, that's good. And then for figure 4, would anybody like?
Hold on. So what was the point of that, though? What's the point of figure 3?
It kind of just showed like how it should, like for a regular sleep, like someone who is not deprived of sleep, how over time, eventually, like the tracer would be cleared out of their system, and yeah.
So it's just
It kind of shows like the control.
okay. That was kind of what I was thinking too. I hesitate to call it a control, but it's almost like it's a test of long-term damage or health of the brain after this procedure, because there's always the possibility that you do this type of procedure and without realizing it, you've damaged the brain forever, and then you never go back to normal, but you wouldn't know that because you never tested it again,
Yeah.
So I think that's the purpose. All right, keep going.
Okay. Would anybody like to do figure 4?
I can do it. Okay, so this is basically just trend plots of percentage change in single unit ratio, and they're indicating the tracer enrichment within the brain tissue that are they're presented at the different regions. A is the CSF space at the craniocervical junction. B is the cerebral cerebral cortex, and C is the cerebral white matter. So, one second. A shows that within the CSF spaces, there was no difference between the tracer enrichment. B is showing that sleep deprivation group had more and higher tracer levels in the 24 to 48 hour than the sleep group. And this shows that clearance was reduced within the cerebral cortex of the sleep deprivation group. And in C, the white matter clearance of the tracer was impaired after 48 hours in the sleep deprivation group.
Without going into the supplement or rehashing old topics that like we did last week, I'm going to just go and say that it looks like they did the statistics correctly for this one. I know we're not used to that, but I think it's correct. For those of you who are wondering, the cisterna magna is actually a big area of cerebrospinal fluid right under the cerebellum. So Shannon, can you go back one figure? If you look on the 10-minute sagittal view, so the top row, second column, 10-minute view, you'll see that the reddest portions are actually around the cerebellum, because these injections were done, I guess, not into vein like they usually do. I guess they did them actually into their as a lumbar puncture. These people must have been paid handsomely for this, because that is not a pleasant procedure. So that's why you see that light up first, because the stuff that's touching the major cisterns, or where the sinuses are that are full of fluid, are actually getting it first. Okay, keep going.
Okay. So figure 5 is similar to like figure 4. It shows the tracer levels in different sections of the area of the brain regions. So overall, like each section is for different parts of the brain. Like each graph shows different parts of the brain. And overall, it shows that clearance was impaired in each of those sections, normally between 24 to 48 hours in the sleep deprived group. So like the frontal cortex, the clearance was impaired in the frontal cortex at 24 hours and 48 hours of the sleep deprived group in A. B showed temporal cortex, that the clearance was impaired. C shows the clearance was impaired in the parietal cortex of the sleep deprived group. And then like D was the insular cortex, E was the thalamus, and F was the amygdala. So each region their clearance was impaired because of being sleep deprived.
And if you know the function of all of those things, you can start to see why people who are sleep deprived have certain symptoms, right? They get agitated, they have trouble making memories, they're emotional. So like D, insular cortex, their emotions. F, amygdala, much more stressed or fearful. E, thalamus, bringing signals in from the body or from any sensory area into the brain for processing and sending them out, also inhibited. The parietal cortex, which is just basically your sensory cortex, and your frontal cortex, which is your decision-making cortex. All of that is impaired in this. Yeah.
And then the final figure just shows the positive correlation between tracer enrichment and the cerebral cortex and white matter. So the graphs show the association between tracer enrichment within the cerebral cortex and the cerebral white matter at different time points. So at A, it's between 4 and 7 hours. B, it's at 24 hours, and C, it's at 48 hours after being the intrathecal CSF tracer administration. So at all points in time, there are highly significant correlations between the tracer enrichment, and it shows the best line of fit and the Pearson correlation of coefficients, which is the R, which are significant p-values.
Now say it with me. Correlation is not causation. Ready? Dear Lord, thank you for allowing us to understand that correlation is not causation. However, when you see it this tightly, when you see an R value that high, there's a pretty good chance, right? Can you go back up one, Shannon? Because I think this was the most important part of, or at least to me, the most interesting part of the paper was the timing. So they were only sleep deprived one night, correct? Total sleep deprivation for one night only.
Yes.
And yet you see that the deficits persist even 48 hours later. They all seem to have rectified after 4 weeks, but 2 days later, even with regular sleep then reestablished, you still see a deficit.
Yep.
We have known for a long time that sleep deprivation is really bad because you don't make up for it. Like a lot of you, I'm guilty of this myself, like, oh, I slept late on Saturday to kind of make up for how little sleep I got in the week. It does not work like that. And this is now really kind of the first molecular evidence in a human that that's the case. It does not work like that. You pull an all-nighter, you're going to suffer with it for a while, and you're going to have all the consequences. Like I always talk to, it's mostly like freshman boys who, you know, they're little tiny people, right? And they want to get taller, but they're up all night playing whatever video game it is that little boys play nowadays. And they're pushing off all of their growth hormones and stuff. And you think, oh, well, I'll just stay up one night. But that growth hormone is now inhibited for at least 2 days after the deprivation. The more you sleep deprive yourself, the less you will grow. And I'm just using growth as an example. It affects a whole bunch of other things. Go ahead, Shannon.
Okay, so conclusions and implications. So overall, the research shows that one night of total sleep deprivation impairs molecular clearance from the human brain, and an effect not compensated for another night's sleep, like what Dr. Tar was saying. The results support the hypothesis that the interstitial space increases in the sleeping human brain. And then it has multiple implications. It could be implicated for understanding the impact of disturbed sleep in the involvement of neurodegenerative diseases, and may point to avenues for enhancing endogenous molecular transport and delivery of intrathecal drugs.
Can we all agree that intrathecal drugs sounds like a terrible idea?
Yeah.
Who wants a needle stuck in their lumbar spine?
Nobody,
no. And also, so like freshmen, when we talk about diffusion and we talk about hypotonic solutions, this kind of is showing that as it's clearing out junk through the night, you're actually increasing the amount of you can think of it as fresh water in the area, so that the poisons, the toxins, the metabolites that have been building up all day are no longer inhibiting further function or poisoning the brain or anything like that. So you're seeing a flow from high concentration poison to lower concentration as it's being flushed out through sleep. Cool. Do you have anything else, Shannon?
No, I covered it. Oh, Johan, you have a question?
I was going through the paper, right? And I was looking at the demographics. And I don't know if this is like based on like what was available at the time, but I see that most of the test subjects are female. Is that like a selection thing, or is that just like an availability thing? Like,
Females can handle the lumbar pain.
How dare you? How dare you? Females don't know what pain is. That's not true. That's a great point. I didn't even notice that. You know, when you write grants to do human work, I was actually talking to Lena Mack about this yesterday. she was asking if I have experience writing grants for clinical work, and I do, but not the type she's talking about. She was talking about farm stuff. In those grants, if you're working on humans, there is a special section of the grant that you have to fill out about how you're going to have equal representation between males and females. And this can get a little odd when you are studying, I don't know, testicular cancer or something, right? How do you get females involved in a study on something they don't have, right? And here you have the opposite problem. 15-2 and 6-1. Oh,
Would this just be a thing of convenience? When
yeah,
do females,
yeah. They're
When do-
already in the hospital.
yeah, when do females typically get lumbar
No.
injections?
I thought that too, but that's not actually. So, well, I'll let the kids answer that. Why do they usually get lumbar injections? Seriously?
Come on, guys.
Ladies, if you don't know this, this is a problem.
It's before you poop on the table. Come on. Come on. Come on.
Pregnancy?
Yes.
Thank you, male. Yeah. Represent. Yeah. The guys know about that and the girls don't. That's how I interpret that. Except that, so I thought that too, Miss Liff, but it's saying no. It's so 5 of the patients total have non-verified CSF leakage, which usually is a cribiform fracture, right in the middle of the skull. Pineal cyst is 6 of them. Arachnoid cyst 2. Hydrocephalus 1. And whatever the heck IH is. I don't know what that is. Non-surgery and surgery is another group. So these people apparently were already undergoing a bunch of tests and surgical procedures anyway. So they were the ones available to do that, I guess. I don't know why it would break down that way, but they tried to keep it as age and gender matched as possible. That's what they said. This is a problem that you run into with any human study. With mice, they're all clones of each other. A C57 black 6J mouse is the same whether it was born in your facility or in Japan. It's the same mouse, same genes. It's a clone. So you can take 100 of them and actually and study this all you want. But when you have humans, it's kind of harder to do. Huh. So I don't know the answer, Johan. That's a good point. I don't know. Another thing I wanted to bring up was that the utility of sleep deprivation anyway. So I just railed against sleep deprivation as this thing that's really bad and you want to avoid it because of, you won't grow or you'll have trouble learning and blah, blah, blah. Okay. Shannon, can you stop sharing your screen? Thanks. We know that people who suffer from depression, if you sleep deprive them, it has a profound antidepressant effect. So sleep deprivation in people with major depressive disorder, okay, will feel so much better the next couple days. And then it'll slowly start to come back. But because of the potential damage that sleep deprivation does, you don't want to use that as a therapy. But there's also another complication. Sometimes it's hard to tell which depressive disorder you have. So major depression is one thing, but if you have bipolar disorder, people with bipolar disorder, the worst thing you can do to them is sleep deprivation because it has essentially the same effect that it does in depression, except it's magnified. So instead of being a relief of depressive symptoms, it ends up being a full shove into mania. And so then all of a sudden people with bipolar, especially type 1 bipolar disorder, will become hypermanic. They will go out and spend all their money. They'll think that they're going to rule the world. They'll go out and party and like it's 1999. It would be amazing. You don't want to do that. Very, very bad bipolar disorder and sleep deprivation. Which is interesting because what about this process is interacting with either bipolar disorder or depression? Anybody want to speculate for funsies? No. How do you treat depression?
Antidepressants.
Sure. Or puppies. Puppies work too. But antidepressants. And what system does antidepressants work on? Which brain chemical?
Dopamine.
A little bit. I'll give you a little
Serotonin.
bit. It's mostly serotonin. Yeah. Mostly serotonin. Excuse me. Mostly serotonin. Staying up means that you are usually it means that you have light signal coming in, and that light signal is it is stimulating the part of your brain, the part of your hypothalamus that releases a chemical called vasoactive intestinal polypeptide. And VIP, as it's called, VIP actually inhibits serotonin release. But if you stay up for a very long time, you run out of VIP, which means you can't inhibit serotonin anymore. And therefore you relieve the inhibition of serotonin, and serotonin then has a more profound effect on your brain, just like an antidepressant does. And all of this happens in this little nucleus called the suprachiasmatic nuclei of the hypothalamus, which you've all heard of, because that's where your circadian clock is. Everything comes back to circadian rhythm, people. Everything. Let me do one plug for Mary Carskaden's work. If you look her up, I'm in danger of doing that usual thing where I talk about a famous scientist and then something bad happens like 2 days later. Mary Carskaden, great woman. If any of you have ever had a sleep test, she's the one who invented that sleep test, the MSLT. She does a world-renowned summer fellowship for undergraduates. So anybody who is anybody in the sleep field has had some training by her at some point. If you like the sleep field, you should consider at some point during your undergrad years doing this fellowship. It's called the William Dement Fellowship. So like William Dement was the guy in whose lab she invented the MSLT, but he also invented a whole bunch of other sleep indicators. Any questions, comments, concerns, criticisms? All right. Shannon, should that paper have been published in Brain, which has an impact factor of 14?
Yes, I think so, yeah.
Is Alex here?
I don't think he is now.
I don't think he is. Is it better than Alex's paper last week? Is this even like a question? I think it's leaps and bounds better than last week's paper. And not to say that Alex did anything wrong. It's good to pick a bad paper once in a while, but this paper was really good. I think really good. Yeah. Okay. Questions, comments? Anybody have anything? No? You all seem kind of dead.
I think this paper could be really useful in job and workplace hour regulations.
Yeah. Well, Dr. Karen Gamble and I did a study, when was that? 8 years ago, I think, something like that, something like 8 years ago, where we actually studied a cohort of nurses go through shift work and saw how devastating it was when they didn't have a regular sleep schedule, like how much caffeine they consumed, how many antidepressants they had to take. It was pretty interesting. They use it it's in textbooks now.
I mean, in one 30-day trip, I've seen one guy hallucinate, totally hallucinate, go crazy. So
I missed the beginning of that. I'm sorry. In one 30-day trip. What's that mean?
out to sea trip. So like
yeah.
20 days of nonstop fishing, of minimal sleep, maybe 2 hours a night.
What, did he do that because he wanted to, or because he had trouble sleeping?
No, that's the job.
Oh. Do any of you have trouble sleeping? No. Must be nice. Okay. Let's see what else. All right. So once again, we're gonna do, I think, 2 tea times in a row next 2 weeks. I think both of them are on Tuesday, 7:00. I really want you all there, and I want you to be talkative, because there's nothing worse than having a guest and then embarrassing the program by not actually participating. So you'll like Lena a lot next week. She's really easy to talk to. You can imagine Brooke Morin, just older. She's just like, Brooke is just like Lena. That's kind of one of the reasons that I hired her as a TA last year. In fact, when they met each other, Brooke was speechless. And then, in the middle of it, I go, Brooke, what are you thinking? She's like, oh my God, it's like talking to me. So if you know Brooke, then you'll kind of understand where I'm coming from. So be active and, ask good questions. And then the following week, I actually don't know him very well. I don't know him at all. Dr. Billick, but his sister is Brendan's mom, and she was kind enough to kind of arrange it for us. And did your sister respond, Miss Liff?
I did.
Uh-huh.
She did. March 7th.
March 7th. Okay, so we'll have an Intel engineer,
Dr. Liff.
Dr. Liff, on March 7th. Good. Okay. It's all shaping up. Things are good. All right. Good job, Shannon. Yay. Yay. You put a little more effort in, Sam. Yeah. Thanks. Okay. And we're good. If anybody needs to talk to me, I can stay on, but thanks, Shannon. Good job. Goodbye.
Thank you. Have a good night.
Thank you.
All right, Dr. Char. See you.
See you later.